Hello, everyone, and welcome to the next installment of the OpenWorm Journal Club. Really excited to have you join us today. Today we have a great session talking about human brain emulation. My name is Stephen Larson. I'll be hosting today. So I'm a co-founder of the OpenWorm project, and we frequently get asked in the OpenWorm projects. Our purpose is creating the world's first digital organism in a computer, and we're very interested in the neuroscience. We very frequently get asked about the prospects for the human brain, and when are you going to tackle the human brain? And of course, there's a lot of great projects that address this question. And so I had the pleasure of meeting Randal Koene, our speaker today, about 5 or 6 months ago. And this is a subject that he is very passionate about. And he is the co-founder of carboncopies.org that looks at this question. And he has also brought along a few folks today to help in this presentation. So we will be hearing from him. So we have an illustrious panel here today to talk about this topic. And before we get started, I would like to introduce them to you here so that you will get a sense. And panelists, thank you very much for joining us. The format today will be that we will start off with Randal making a presentation, and if panelists have any questions, they are free to interject during the course of the presentation. And if not, we will have a question and answer session towards the end. Now, if you're joining us online, please feel free to ask any questions via social media. You can ask us, you can reach out via Twitter, Facebook. You can ask us via the Google Plus event page. And possibly, if there are enabled YouTube comments, you can do that way. Please also, if you are watching us on YouTube, you can feel free to subscribe to the channel that's here. So, okay, so without further ado, let's introduce our panelists. Looks like folks have just arrived, so this is great. So we're very excited. So actually, let's go ahead and start with Chester. Chester, just joining us here. Please go ahead and introduce yourself, say a little bit about yourself and where you are joining us from.
Okay, good. Good day. Chester Grant. Joining you from Antigua, that's in the Caribbean. A lecturer at Antigua State College. I'm the creator of a artificial general intelligence group. I'm mainly taking my stance from a more theoretical part of computer science. I am not really familiar with neuroscience, but it's something I'm interested in terms of applying it to artificial general intelligence.
Okay, great. Thanks for joining us. And so we will probably be muting folks just if they're not speaking. So if you can just stay muted when you're not talking, then we'll not have background noise. But thank you very much for joining us, and hang on to some good questions here as we go forward. So let's go to Ben Shi.
Hey everyone, my name's Ben. I'm a PhD student at UC San Diego in mechanical engineering, and I'm working on soft robotics. So I also don't have much neuroscience background, but I'm interested in the fabrication and manufacturing side of soft sensors, and I'd like to incorporate neuroscience into my projects.
Great. Thanks for joining us. All right, let's go to Kim Reinhold.
Hi guys. I am a recently graduated PhD student at UC San Diego as well, and I'm a neuroscientist. I've been doing experimental neurobiology recording from the brains of mice, the visual system specifically, and using optogenetics to manipulate cortex and thalamus and look at how that affects processing.
Terrific. Thanks for joining. Okay, and Phil Rhodes.
Yeah, how you going? Yeah, my background is originally in genetics and zoology, more recently in population genetics, but the main reason I'm here, I guess, is because I'm involved with an organisation in Australia called the Neural Archives Foundation, which is somewhat of a competitor with carbon copies, I guess. Although there's no competition amongst the friends, I guess. And also I'm the Executive Officer of the Cryonics Association of Australia, and there's a couple of other general interests as well.
Great. Thank you very much for joining us. All right, going to next panelist on my screen looks like Tim Mullen.
Hi there. So I'm a computational neuroscientist, and my research has primarily focused on large-scale dynamical modeling of human brains through electrophysiological techniques. I also have a background in computer science and always had a bit of a passion for artificial intelligence. I'm CEO of Syntropy. We decode brain states on the cloud, and I direct a research lab there as well. Glad to be here.
Thanks very much for joining us. All right, and Tom Porteges.
Hi, hi everybody. I've been associated with the OpenWorm project for a couple of years now, probably going on 2.5 years. Of lately, I'm kind of hooked up with another sort of affiliated group called DevoWorm, been working on some projects associated with that. My background is computer science, artificial intelligence, machine learning, so I'm in that sort of group. But I'm really interested in the biological and neuroscience aspects of and what can be obtained toward the computer science area. Thank you.
All right, great. So as you can see, I think we've got a great mix of folks who are, on the biology side, on the neuroscience side, as well as on the computer science side, so I'm hoping for a really robust conversation here. Obviously, feel free as questions, come up to interject and ask them. All right, so our last three folks in here are from Carbon Copies, so I left them to be introduced at the last. So let's go to Stephen.
Hello, my name is Stephen Fry, and I studied neuroscience at Berkeley. I have a long-standing interest in neurotechnologies. I founded the Cognitive Technology Group at Berkeley. In the past, we created an interactive laboratory of brain technologies at the Exploratorium Museum in San Francisco. We also have a blog on varieties of brain technologies and cognitive enhancement. And I'm very excited to see how the ecosystem of technologies that Randal and other people here have done to build out our tools for understanding the brain.
Terrific. Thanks for joining. And let's go to Keith.
Hello. My name is Keith Wiley. I'm a fellow with the Brain Preservation Foundation and the author of A Taxonomy and Metaphysics of Mind Uploading, which I came out with last year. I've known Randal for over 20 years and have watched how he has maintained and developed the Carbon Copies project, and I try to assist him with that as much as possible. My formal background is computer science. My day job is sort of a big data analytics and machine learning, but actually my writing in the topic of mind uploading has been far more on the philosophical side of how we interpret the nature of personal identity and minds and consciousness in the context of these sort of hypothetical substrate independent procedures which may actually come to fruition in the future. So I sort of dabble all over the place, I suppose.
Terrific. Alrighty, and then last but not least, we will now go to Randal. I will be sharing his presentation. So Randal, after you introduce yourself, just feel free to say, let's go into the slides, and just tell me, next slide, and I will iterate them for you. So, Randal, please.
Sounds great, and thanks for having me on. So as you know already, I'm Randal Koene. My background actually started in physics, and then I went into electrical engineering to learn about information theory and on to neuroscience, knowing that I was most interested right from the start in how to do human whole brain emulation, or leading up to what's popularly known as mind uploading. It's really interesting to see Philip here from the Neural Archives. I think I found out about them first when I was in Melbourne a couple of years ago, I don't think that there is any kind of competition as such between carbon copies and neural archives. If anyone is going to be a competition there, maybe it's the people who are doing brain backups or something like that back out there in New York. What I do is more on the level of trying to create projects, putting the problem people together with the solution people. That's really my side of it, so the road mapping and getting people to work together. And with that, let's get right into the presentation.
Okay,
there it is. So I'm going to try to be pretty quick about this. The main reason for that is because I have a hard stop at 10:30. I have to jump to another important work meeting that's going on. And so I'm going to go through this fairly quickly with the assumption that even if you're not all neuroscientists, you're generally scientists or engineers, and so the technical stuff isn't that hard for you to understand. I'm going to talk about this generalized roadmap. How can you get to whole brain emulation? I'm going to talk about some details such as system identification and some tools that are involved. And please go on to the next slide. I won't explain exactly what these steps are, except to say that there are four main parts to this. Let's go straight into the next slide. Okay, so something really basic here. The exact sciences really depend on improving understanding by describing observed effects through representations and models. Some things about nature are predictable, and pieces of nature exist within an environment. The various pieces aren't really independent. Conditions at some piece in nature at some time predict aspects and conditions in another piece somewhere. We say they affect each other. And there are signals between those pieces that convey the information. So we want to understand more about the predictable dependencies. So we explicitly describe the signals and how the information they convey is processed. Next slide, please. Now, of course, nature has an awful lot of pieces, and descriptions become really complicated. A systematic and iterative improvement of a description is what we call model building. And initially, of course, we try to keep things simple. We always try to build a simple model first. We constrain our models. There is some effect that's of particular interest, and ideally we focus entirely on the scope and details that are needed to explain that effect. In neuroscience, the interesting effects are often called task-specific behaviors. So, for example, object recognition, emotional responses, executive decision-making, and even consciousness or aware behavior. Next slide, please. Now, we know that our pieces of interest, for example, a molecule or gas or a neural circuit in the brain, we know that there are certain pieces we're most interested in, and we try to look at how they may be communicating with others. So what are the signals that could be involved in these effects? Overall, physics describes things, of course, ultimately in just a few major types, like the big ones, gravity, electromagnetism, a weak nuclear force, and strong nuclear force. Of course, those aren't the ones we're really going to be working with. We can constrain it further to things like electric current, electromagnetic radiation, and so forth. So a piece of neural tissue may be responding to an ionic electric current, temperature gradients, pressure or shearing forces, somatic transmission, electromagnetic fields, and many more things. And experimental work helps us to create a priority order to see which ones of those really are most involved in the kinds of activity that we're interested in. By and large, most of those signals disappear and drown in the noise. They lose their predictive value. Electric currents, and in particularly those powerful discharges that we know as neural action potentials, which unfortunately are not the typical thing you see in the C. elegans worm, or spikes, appear to carry the dominant information in the mammalian brain. Next, please. Now, in control theory, the piece of nature that you're modeling is sometimes called a black box, which has a state, and it receives input and it produces output. The process of updating its state and generating output is described mathematically by some transfer function. If we find a suitable transfer function, we learn about that black box by looking at the context of the behavior and the signals. Now, there are some formal methods for figuring out what this transfer function can be. One example that's very general is to try to find the kernels of a system expressed in what's called Volterra series expansion. I'm not going to get into that detail here. Next. Okay, we can skip right over to the next one. So the effects that we're interested in are those that we would associate with our experiences when we're talking about whole brain emulation. sensory perception, learning, memory, problem solving, goal-directed decision making, emotional responses, consciousness, self-awareness, language comprehension, and also motor control and things like that. Some of those are externally observable, and some of them are parts of our internal experience. Neurophysiologically, they involve the interactions of ensembles of neurons with a specific circuit layout. So let's go on to the next slide. There isn't really a consensus about exactly which signals are or are not the essential ones for brain function, but we can take an iterative and systematic approach here. We make some initial assumptions about the signal-to-noise ratios, about the kind of output that reliably affects the environment during interesting behavior, and about the sort of signals that neurons are well suited to deal with. Biophysical mechanisms of sensory input, for example, at the cochlea or the retina, produce electric nerve signals that are characterized by trains of fairly uniform neural spikes with very specific rates and time intervals. Similarly, the primary output through muscle control, for example through your vocal cords, also employs trains of neural spikes. And finally, a primary means for long-term state change, learning, is governed by modified synaptic strength, and that modification also depends crucially on the temporal order and time separation between pre- and postsynaptic neural spikes. So a representation that successfully predicts spike times may be a good first iteration for a model of system processes in neural circuitry. You can kind of see how that would make sense. And there's one case where some really interesting results were already achieved using these assumptions and demonstrated in what's known as a cognitive neural prosthesis that was developed by the lab of Theodore Berger at UCS. So using system identification in Volterra series expansion, their team developed a chip that contains a multi-input, multi-output model with nonlinear parameters that are specified after learning from consecutive presentations of spike data. The input of the system is obtained from an array of electrodes in a region of the hippocampus known as CA3, while the output is delivered to a region known as CA1. Those regions happen to be crucial in the formation of new declarative or episodic memories. And that chip can be used to alleviate dysfunctions of that area. So they're thinking, for example, the fields of stroke or trauma or some other disease. There's also a more general technique that was designed fairly recently to work out initial assumptions. It was developed by Lazar and Slutsky, and it's called the development of channel identification machines. It's a formal method for identifying a channel modeled as a multidimensional filter in a system where a communication channel is cascaded with an asynchronous sampler, such as, for example, an integrate and fire neuron. And a channel can be approximated to any degree of precision, and that method is therefore completely generalizable and can be applied in noisy conditions. But again, this is not something we have to get into detail about. The next slide, please. Okay, right on to the next. Okay, so meaningful system identification that could reproduce both observable behavior and internal experiences or experiential states of an entire brain is completely unfeasible, of course, when the whole system is treated as a black box, if we're looking at something the size of a brain. It has to be broken down into many black boxes that communicate with one another. So you need to choose your smaller black boxes, their size, their location. You need to acquire enough data about the input-output correlations at those small black boxes so that you can do system identification for them. And you need to understand the relevant communication that's possible between the various black boxes that are there. Now, if you go to the next slide, and sorry about those cluttered slides, by the way. This is an amalgamation a bit of a presentation that I did at UC Berkeley a while ago, and for some reason there were many more visuals in there.
No worries.
Okay, so now we're getting to a roadmap. Really, we can address the interaction between the subsystems by looking inside and noting the locations of the smaller components and tracing the connectivity between them. So for spike trains, the communication pathways are dendrites and axons, and the synapses where they meet. And it's the field known as connectomics, of course, that deals with that problem. And I'm sure you're all very familiar with that, or have at least heard of it in the context of, say, the connectome of the C. elegans. For other effects, like the extracellular field potentials and diffuse neurotransmitters, the surrounding medium and emissions and diffusion, they can all be taken into account A well-known choice for these individual black boxes that contain very tractable subsystems is decomposition of neurons into what's known as an electrical compartment, analogs of so-called compartmental neuromodeling. The input-output data that can be obtained largely determines if this or another level, such as whole neurons, is the appropriate level of simplification. High-resolution connectomics by electron microscopy obtains the morphological data for compartmental modeling, and there's also another method of microscopy now that I'll get into in a bit. There are a number of labs that are working on that, and back in 2011, this approach actually led to an interesting proof of concept that was published by Brigman et al. And also another one by Bach et al. Using data from the Winfried-Denk lab. And I think I'll get into that a bit more as well in a moment. To properly characterize the responses of a neuron, we need observations that allow us to set and test values, parameter values that relate to the sensitivity and manner in which input currents affect the neural membrane potential and the resulting action potential when a threshold is reached, as well as the time course of restorative dynamics, so after hyperpolarization, after depolarization. Brain emulation is actually closely related to neuroprosthetics or neuroprosthesis, which again is closely related to neural interfacing or brain-machine interfaces. It's kind of a blossoming field in neuromedicine. Building neuroprostheses requires many of the same steps as brain emulation because you need to carry out the system identification on the piece to be replaced or supported. So good neural prostheses are in effect brain piece emulations. Both brain piece and whole brain emulations have the requirements outlined in this roadmap. And collecting the measurements needed means that you have to have an interface with the brain, and that's the field of neural interfacing. So you can see how it all kind of overlaps there. Next, please. Okay, so let's get into some of the tools for parts of this roadmap. In neural tissue, sensible boundaries have to be drawn somewhere around these pieces of the neural circuitry so that you can look at those I/O contacts between the different pieces and identify them. A geometric decomposition into 3D stacks of voxels of equal size is one approach. That's what you could do, for example, with an MRI, although an MRI is perhaps the wrong resolution for some of the things we do here. Another method is to identify neural cell bodies, like in a slice or in a culture, on top of an array of electrodes, and to use the correlations between the measured activity at each cell body to derive a functional connectivity map. And then there are some variations of each of these, such as Anthony Zador. He's developing a biological protocol to derive the target neurons of any neuron. So in the case of Zador's method here, he uses biological markers, or is planning to use biological markers such as a unique sequence of RNA or DNA, synthetic RNA or DNA, to mark the pre- and postsynaptic sites of synapses. Those markers then act as bidirectional pointers between neurons. It's kind of like you're sending postcards to all of your neighbors. But the most detailed and successful tools to date are really those where you have to section or ablate pieces of brain tissue and take electron micrographs at a resolution of about 5 nanometers, and then you build three-dimensional geometric morphology back from that. And with that, you can identify even individual synapses, and there have been some really excellent results. Next, please. So system identification for individual neural compartments can use standard models that employ an electric cable analogy and Hodgkin-Huxley equations. The morphology can provide some insights into functional behavior. For example, compartment radius and length constrains the conductance of electrical currents. Morphology can also categorize a neuron or synapse, which constrains the possible response functions. Despite those constraints, even small systems contain numerous parameters, and not all of those relate directly to visible and unique morphological features. Even where they do, the reliability and precision of measurements is not always adequate. Next, please. So parameters have to be tuned such that subsystems behave sensibly on their own or in cohesion with connected neighbors. And we can do system identification for signals of interest at a black box by observing activity, or when we call this a gray box, by also being able to stimulate and observe, so then you can set up whatever signals you want and watch what's happening. Tuning and verification involves measurements at these reference points. Now, if the resolution of reference points is less than the resolution of structural decomposition in this model that you created, then system identification depends on our ability to map measurements to a collection of subsystems and the combinatorial size of that problem. So in how many ways can the subsystem be interacting to produce observed responses? We may not be able to determine system parameters uniquely if that number is large. The amount of observations needed and the duration of observation increase with complexity. Clearly then, there's great value in having tools that provide measurements at many more reference points, ideally at a resolution that approaches the resolution of those subsystems you're interested in. Next, please. So this is the problem of those high-resolution functional ways of recording data and being able to do the system identification. Now there's a really strong interest among neuroscientists in the development of these tools. And you can look at labs such as the one of Stan Skafidas that are exploring the use of nanoparticles as signal enhancers for traditional techniques such as MRI and CT scans. There are also arrays of thousands of recording electrodes that are being developed and combined with optogenetic techniques so that selective observation of specific groups of neurons can be guaranteed. And there are microscopic wireless probes and functionalized nanoparticles with simplified task-specific capabilities that are being developed to counter some of the disadvantages of the extensive tethered electrodes. And there's a collaborative effort underway to create biological tools that employ DNA amplification as a means to write events onto a molecular ticker tape, as it's kind of called. The project goal is to be able to record signals from all of the neurons in the brain and potentially to measure at resolutions beyond that. And you can see how a molecular technique can be useful there because it's very easy to get that distributed over all of the cells that are there, over all of the neurons. Okay, detection of neural activity in many different signal domains, like, say, all of the different pieces of the physical universe. Neurons respond in some manner to every type of impinging physical signal. This is the thing you have to realize, of course, and the internal operations also influence all types of signals. So there's so many different kinds of signals you can look at to see what is the best one to use. Is it electromagnetism? Is it ultrasound? And optogenetics has already shown that some modification can make responses much more pronounced. And there are similar investigations that are interrogating techniques on the recording side. There may be many other avenues to go along in this. So it's really a broad sort of out-of-the-box thinking to try to come up with better ways to record and to stimulate. Next, please. Some challenges are very general to system identification and all across whatever it is that you're trying to identify, and some are particular to neurons and neural models. And by the way, that's one more slide, please. Next slide. It's not this one. There's some unique challenges that arise when you're working with pieces of neural tissue and consequent large neural circuit models. And some challenges are exclusive to the domain of whole brain circuit reconstruction. Many of those involve the integration of techniques for data acquisition from structure and function that are developed with the constraints of particular novel tools. Next, please. A careful assessment of system identification for the experiences we wish to represent demands that we consider contributions outside the domain of neural spiking. For example, are the experiences meaningfully represented by states of cells other than neurons, glia perhaps? Or are there significant ways in which neurons influence each other even in the absence of spiking? Can neurons relate to each other without receiving spikes or activity directly caused by spikes? And if you're working in C. elegans, then this is an obvious thing for you. Our initial assumption is that predicting spiking within an acceptable error range implies good emulation. And spikes are not epiphenomenal, but rather the currency upon which sensation rests. Spikes precede ensemble responses and field emissions, so there's some sense in that. An important challenge is, of course, to test those assumptions. Next, please. Good temporal spike predictors demand that we observe or deduce when spikes would occur in the original system. Additional information, like membrane potentials and influences on those, can improve our ability to make those good predictions. Next, please. Okay. Snapshots of baseline or differential activity in large volumes of tissue that are provided by MRI are often too imprecise for parameter tuning, but they can be a means of model validation. The model should produce a sensible virtual MRI, once you've created it, in terms of distribution and propagation of activity. The challenge is that aligning a virtual MRI generated by the model with actual data would demand that the model also replicates the expected 3D spatial geometry. Next, please. Detailed geometrical and morphological data is provided by 3D reconstructions from EM scans, as I already mentioned. And there's one way to try to deal with that, then, is to say there's a correspondence between this morphology and function. Often there is, it's not a very clear picture of how you would do this, but there is some idea that it may be sufficient to look at the morphology and to be able to deduce the function from that if you know enough about what all of the different cells are and how they work. We can look at the morphology of a cell and we can tell if it's a pyramidal cell or if it's an interneuron, and that helps us figure out what kind of a model we would use, what would be the activity dynamics for that, the receptor channels that are likely to be present. Still, this component identification is challenging because the classification currently relies entirely on morphology, at least when you're using EM. There's an effort to add direct means of protein identification, probably using different microscopy techniques, that would alleviate that problem. So when you reconstruct in terms of compartments, using radius and length of a cylinder, giving you an estimate of the resistance and capacitance involved, even though those estimates also depend on the model of identified type of neuron, those measurements are subject to a degree of reliability. You can average, but that doesn't remove the cumulative systematic errors, so there are other issues to consider there when you're trying to rebuild this from morphology. Some of the measurement data is also likely to be very hard to recover because of errors just in your ability to scan everything properly. Right now, also, the tools that are being used in labs like the one by Winfried Denk can only handle very small volumes of neural tissue. Large volumes are going to require an automation of sample handling, which is something that, for example, Ken Hayworth is trying to do at Genelia Farms. Okay, let's go on to the next slide, please. Now, there is an entirely new approach to microscopy that's kind of a result of out-of-the-box thinking that happened at the Boynton lab, where the idea was, well, if we're trying to see more, if we want to get to a higher resolution, we want to see a lot of things and also a big volume, making a bigger microscope might not be the right idea. Perhaps instead you can make the sample larger. And that was achieved with this so-called expansion microscopy, which uses a paraformaldehyde procedure to expand cells up to 20 times. The nice result of that is that not only do things become visible that previously required electron microscopy, it also means that errors, say for example, problems from cutting the sample, become, relatively speaking, 20 times smaller in the result after you've done your imaging. Now they're combining this procedure with molecular barcoding techniques from the Church lab, which are similar, by the way, to the stuff I was talking about earlier with the molecular ticker tape, as a means of tagging proteins in cells using synthetic pieces of RNA and DNA. And then those tags can be read out using what's called in situ DNA sequencing, a new process for doing sequencing in a slice. Then you can wash out the tags and put in new tags again and do that over and over again in a sequence, so you can see a lot of different things in the same sample. This is a huge advantage to using optical microscopy. Next, please. Now, brains are, of course, plastic. And mostly we think of plasticity in terms of learning the modification of synapses and even the availability of connections. But there are other kinds of plasticity that we often don't think of, namely in terms of deformation. Apparently, some aspects of the morphology are relevant to model building, while others are mere features of the snapshot that was taken during a scan and then a reconstruction. Present tools for structural connectomics offer little insight into the temporal dynamics of these gradual changes in neural circuitry. Next slide, please. Tools that functionally characterize activity at reference points should give some insight into the temporal dynamics and memory. This ticker tape approach that I started talking about before may even be able to record from all of the neurons simultaneously. Encoding by means of voltage-dependent increases in the error rate of DNA amplification is not entirely reliable, which is compensated by using multiple tape per cell. But before we get into those kinds of reliability issues, let me just quickly try to outline what happens here. The idea is that you get a piece of synthetic DNA or RNA inside of a neuron where it experiences circular amplification, and you have a channel where, as the neuron is activated, as there is more activity, it causes more errors in the amplification. So if you do the DNA readout, the sequencing, and you look at the rate of errors, you get an idea of what the activity was. But there are all kinds of problems like this. This reliability, but also how long can the recording be? How fast is the rate of recording? Is that enough to capture all of the spikes that are there? Leads to having to have multiple of these tapes in every cell, and these are things that the George Church Lab is trying to fix right now to figure out how good can they make this molecular ticker tape, which has the advantage that if it works, you can have it delivered to all of the neurons at the same time. Next, please. There are also some challenges if you're trying to use functional data obtained by fluorescent microscopy, which is another very popular approach. Using those tools to obtain full coverage throughout the neural tissue and in complete brains involves significant disturbance because you have to make viewports and the insertion of microscope devices to be able to see what's in there. So it really works best with very small pieces or with very small animals. And this is kind of a problem that's similar to one that's faced by using large electrode arrays. Next, please. But there is also another interesting thing being developed right now, microscopic wireless electrodes that are a feasible alternative where you basically think of every probe as being constrained to a specific task, and the probes having only the pieces they need, like the recording, but not a tether that leads out of the body. An example of this is the neural dust proposal by Maharbiz, Carmena, and Rabai at UC Berkeley. The challenges there are that the power requirements and the demands of data delivery can be difficult for methods like this. They make it difficult to work with a whole network of probes like that without, for example, heating up the brain too much. The group at UC Berkeley tries to get around that by using ultrasound as the medium for both power and for information transfer, because ultrasound isn't absorbed very much by the brain, so the amount of heating is less. Yeah, but that's still very much in development. I think they wanted to test this method in mice in the summer, and I'm not entirely sure if that's happened. I should check. Next slide, please. Now, when we have a way to record spikes, electric field potentials, or membrane potentials from all of the neurons in a piece of neural tissue, we still need to know what is a sufficient sample set and what is the required sample rate for this. So can we predict the neural dynamics from the observation of the shape of an action potential response, for example, or should we observe the response to a collection of possible input combinations in order to estimate connection strengths and predict spike times? Do we need to run the cell through its paces with a full battery of stimulation protocols? Does tractable system identification demand that we do it at a higher resolution on pieces of dendrite? Can we map lower resolution activity data to higher resolution structure data so that compartment parameter values are sufficiently constrained? There are all kinds of problems here with working with the data. Next, please. Many of those challenges that I just listed can be dealt with more confidently if the process of system identification is tested by iteratively building incrementally improved models of small systems. So a small system can be a proof of concept that demonstrates steps of the process and overall feasibility. There are some small systems that are receiving attention at this time. There are, of course, several groups that are working on the nematode, and OpenWorm is a really interesting one. Others are reconstructing pieces of retina. Neuroprosthetic applications are being built for pieces of the hippocampus and for the cerebellum. And there's also a project to extract memory directly from a piece of neural tissue, eventually, or at least from the morphology of it. This is the plan that Sebastian Sung has going at MIT. Now, whatever method we use here, you need to be able to combine both the anatomical and functional measurements, and that's a very significant task that still needs to be completed for even any one of these examples. And then the computational complexity of the system identification ends up depending on the density of the measurement reference points. At some point, what you're really looking for is to try to find this optimum where you go to the maximum that your tools will allow to give you as much resolution as you can, and therefore to simplify your system identification problem. But you need to also go as far as you can in dealing with larger black boxes, more complexity on the computational side, because that relieves you of the necessity to go to higher resolution with your measurements. So it's sort of the sweet spot that you're looking for between tools and computation. Okay, next slide, please. It's often very difficult to gauge which difficulties are going to turn out to be significant problems unless you work your way through the entire process. And that's the main reason why proof of concept systems are so important, like the C. elegans. System identification is obviously not a new field. It's been done in every area of the exact sciences and engineering. Undoubtedly, most of the problems that are encountered when working with neural tissue are not entirely novel either. Some examples of similar problems and the solutions that are employed may be found in other fields. But from the discussion above, or from what I've presented so far, it should be clear that while it's important and useful to build tools that acquire high-resolution structure data and that acquire high-resolution spatial and temporal functional data, that's not the whole solution. Other significant challenges are the integration from different data sources, if you're using different tools, for instance, turning a sea of data into parameter values and validating those values. And so one of the things that really impressed me and intrigues me is this open distributed science model that OpenWorm is pioneering. I'm really curious to see which challenges, for instance, proof of concepts, identifying resolution requirements, or other challenges that are out there could most benefit from that model. And another question that I'm most interested in is also to discover, having seen this landscape of the roadmap of whole brain emulation and the kinds of projects that are involved, are there any areas of overlap that really benefit from direct collaboration? For example, are there any aspects of this challenge that could be considered almost a subtopic or a thread of the OpenWorm project? And I think if you go to the next slide, you'll see that's it. Yeah, thank you very much. And of course, I'm also thanking the people who were involved in all these projects and making all of this great data available. Thank you.
Awesome. Okay. So now we switch over to the discussion period on this. A little bit of feedback from your side, Randal. So if so just be hot on the mute button. So I hope folks in the panel here have come up with some good questions. We also have had a little bit of questions coming in via social media. Just want to sort of pause and let everybody know that we're hearing from Randal Koene from carboncopies.org. We're talking about human brain emulation, and we've got a great panel of folks assembled here from all the way from the computer science side to the neuroscience side. And so I do want to open up for questions. I want to frame what we've heard because I think it's interesting that so a lot of the technologies and research that you're talking about are things which are happening in sort of conventional neuroscience, and in some sense there's really nothing controversial about the tools and techniques. But the way that you're assembling it is something which I think in the field of neuroscience doesn't get talked about as much. Although you can tell us how it's getting talked about more, which is the part where you start to inject the more of the engineering side to actually reproduce what's happening at the whole brain level. At least this is my observation. So as a way to kind of start to bridge this conversation, I guess I'm interested in, as you're thinking about this going forward, how do you see folks in the field of neuroscience, of experimental neuroscience, taking up these ideas, or in any way not taking up these ideas, and how have you found that process?
Yeah, so that speaks more to the how much do individual groups, how much do the individual projects think ahead to this sort of big picture stuff, or how much do they not? And I would say that that falls into, as you said, there are kind of the traditional experimental neuroscientists. There's also this whole side that we would call neural engineering, and then there are neural tool builders. And it's interesting when you look at who seems to be interested in putting together stuff the way that I'm looking at it and kind of putting that engineering angle on it. It's not so much the people who are doing the traditional experimental work. So it's not generally the person who's exploring how a particular receptor responds to, I don't know, cannabinoids or something like that. It's generally either the people who have an engineering challenge, like they want to create a brain-machine interface, or they want to create a prosthetic, or they want to do something of that nature, where they have to pull it together from multiple different directions. They have to work in a multidisciplinary way. Or it's the people who are building tools and they have an interest in mapping out something entirely. So you've got this group over at MIT that really wants to be able to record from every single neuron in the mouse brain at 1 kilohertz resolution. That's sort of their goal, their group goal, the goal of the physics of brain activity mapping group that involves the Boyden lab. And it's because they built the tools to make that happen. That's why they're thinking in that sense.
It seems to me that on this topic, we have a problem where we have a lot of intuition and information in the minds of experimentalists that is not being transferred to those people who are working on the engineering side of things. So for example, when you talk about needing to choose the proper level of temporal and spatial resolution to begin to study or emulate components of the brain, it seems to me that experimentalists might have a deep intuition for that, especially people who have spent their lives studying brain area X or Y. Yet the engineers often approach the problem by trying lots of different things. And I think both are important, but maybe there's a question also, how do we take the information out of the minds of people who have a deep intuition and understanding about specific brain areas, maybe by polls or collaboration, and put that information into the hands of engineers. It seems there are almost two qualitatively distinct approaches to whole brain emulation. One would be to just have an engineering problem and take any possible solution, say do some unsupervised learning to come up with a solution and then try to understand what that solution is doing, versus really understanding how the brain does it and then replicating that. And so I'm kind of curious how you, Randal, and the panel think about these two approaches. Is there any value to, for example, having a huge training set, sticking it into some random neural network, training up that network, and then having that network output something without understanding what's actually happening inside the network, versus needing to understand how the brain works before we can begin to emulate the whole brain? What do people think of these two approaches?
Well, I think those are both valid approaches. It sort of depends on your goal in any given project. So, for example, Tim Berger's hippocampal prosthesis. One might argue that the most important goal of that project was not actually to vastly sort of solidify our understanding of how parts of the hippocampus work. That would be a great goal also, but one might say that the central goal of that project was to systematically model and then prosthetically sort of replicate damaged neural tissue, regardless of how it works. So you take sort of a black box approach to the understanding, which is not to devalue the understanding question. That could be a more central question in other projects, just sort of general science of learning how the brain works. And I would say there's sort of substantial value in all of these approaches. And I definitely agree with, as has already been stated, I think we all agree that the more collaboration we can get going between disparate groups and definitely disparate professionals, neuroscientists and engineers and computer scientists, and maybe a smattering of philosophy. The more collaboration we can get going on these things, the greater our progress will be.
Yeah, I liked very much that Kimberly started out right away thinking about how could you maybe get the information from the experimentalists by using this sort of more distributed system that OpenWorm is really good at, polling data, getting people to pool their information in some way. So I think that's fantastic. And as for which approach is better, I don't think that going to one extreme or the other is necessarily the best, because you can dig deep into the biology of the brain for a very, very long time before you could say we understand the brain and then build your way up again. I think it's more of an optimization or constraint satisfaction problem. You're trying to find the minimum amount of depth that you need to get your problem solved. And if your problem is functional replication at some level, like for the ability to do perception, the ability to do motor action, and all that kind of thing, then you don't necessarily want to go any deeper than you have to accomplish that goal. And yeah, this is still kind of a vague answer, but my answer is try to approach it from both sides and optimize where you're working at by sort of iteratively, quickly getting to that level you need to be at, rather than diving all the way down and figuring everything out at the atomic level and then working your way back up.
That makes sense, yeah. I think we need all approaches. It's just sort of the issue of meeting in the middle. That's an exciting kind of frontier.
So I'm curious, if I may, I'm curious what people think are the central challenges in scaling our current sort of microscopic techniques that work well at very small scale, or we have large spatial resolutions, large scales like MRIs, but which don't achieve the resolutions we need. What do people think are the central challenges, the biggest challenges to sort of scaling this all the way up to larger brains, even whole human brains eventually? We all realize that there will be ongoing challenges, but I'm curious what people think are going to be the things that will make that the most difficult to achieve.
So I have one point on that. So as a computational neuroscientist, my work focuses more on the machine learning, the modeling side of things, trying to make sense of streams of data. And this brings to mind an important part of the problem. Actually, as a short little story, some years ago we had there's something called the Sloan Schwartz Consortium for Computational Neuroscience here, and we had a meeting at Genelia Farms a number of years ago. And they were giving us all their great insight into the process that they're applying to basically model dynamics from Drosophila, map the whole brain, model behavior, etc. But they said, the reason why you guys are here, why we want to talk to you, is because we have no idea what we're going to do with all this data once we record it. And we need computationalists to try to make sense of it. And I think that is probably one of the biggest problems that we're facing right now. So we're coming up with very good ways to record very large amounts of data from the brain, but we still barely know how to process a few sensors of data that would, like 128 channels of electrocorticography data, is so rich with information that we don't know what it means. So for me, that's the biggest challenge from my point of view, is what kinds of computational techniques are we going to have to develop, what kinds of new algorithms, what kind of heuristics are we going to have to come up with that allow us to deal with these massive quantities of data that we're going to be getting from these new sensors. And I'd be very interested to hear any of your guys' thoughts on where you think the quantum leap might be there, other than, of course, more powerful computers, which only gets you so far. Not to mention Moore's law is kind of petering out. So.
Yeah, I definitely second that. In my lab, we're all experimentalists, and we already run up against recording too many neurons simultaneously for what our, servers can handle processing. And so people choose to record less data because they simply can't analyze more data. And so I think it's a common problem in neuroscience. Surely the solution has to be something to do with more computational people with better, algorithmic experience really coming into the field. I think for a long time we've had scientists writing algorithms, which is great as a first step, but sometimes those algorithms are not at the top speed, efficiency, that sort of thing. My hope is we can find some new field. I mean, computational neuroscience is the beginning, but that better addresses these algorithmic problems, which we're really running into in neuroscience.
Yeah, I think the common thread that I'm hearing here from Keith's question to how do we scale up to Tim's point about better algorithms and Tim's point about running into the limitations of data collection is that quantification of where we are now is key to quantification and extrapolation is key to this. And I think that's something I've seen in some of the roadmaps. It was maybe not something that Randal, you focused on in this particular talk, but just this idea of, okay, so if our current capacity to record today looks like X, and we can store spike trains and this much data now, and how many hard drives will it take full of data to capture every single neuron in a human brain, based on how many neurons we expect that there are. Some of that extrapolation can be done today, and some of the numbers that come out are pretty large, but I mean, it's there are other scientific fields that deal with very large numbers. And astrophysics deals with very large numbers. So we can also deal with very large numbers. I think that quantifying some of those things are important. Also, when you get to electron microscopy and you say, well, from the ability to cut up brains and look at them under the electron microscope, as you pointed out, techniques by Denk and such, what is the surface area of tissue that they're looking at now? What is the surface area tissue they would need to look at for a whole brain? Okay, what is that number? How big of a, how much high throughput would we need from an electron microscope exactly, given our current limitations? And then we'd have these really big numbers. And then at that point, I think that's where our engineers start to come in, which is to say, okay, that's a really big number that we have now. How could we pare it down? And it's also where the algorithms come in, which is like, okay, so now we need an algorithm that takes, all the data coming out of EEG recordings and pares it down. Or now we need algorithms that take all the single cell recordings and pares it down. And how could we compress that? And how could we efficiently analyze it? But I think probably the first step is just quantifying like how big the problem is, right now. It's a name of bytes that we don't is like goes off the edge of the yotta bytes, or off the edge of the SI scale right now. But I don't know. I think that would be a practical thing from an engineering perspective to start doing.
So I actually I think that just the question of how much data you need to have or how much data you need to store and collect, yeah, sure, it's a big problem, but it's not the biggest one. I'm actually very much agree with Tim that the biggest problem is the translation. So for example, in some areas, getting something useful, usable parameters out of your data is easier than others because the level at which you're collecting the data is the same level at which you're trying to replicate the system. So for example, in Ted Berger's case, when you're trying to replicate a system at the level of the neural activity and you're recording neural activity, then it's still a hard problem, but it's doable. You can get that data out of the recorded information. But if you're recording the morphology of neurons and you're recording maybe some information about proteins, and from that you want to infer the functional model in terms of electrical activity, then you've got a translation problem. And that's something that needs way more attention, because there's this sort of thinking that if you can just preserve a brain specimen properly and then you can cut it up and you can record a whole ton of this morphological data on it, no problem, we've got the brain, but it's really just a picture. It's not something usable. So that, to me, is the main problem. And then if you're talking about recording electrical activity, the main issue is the size of your black box. Because if your black box is big, if it has a lot of parameters in it, and you're trying to derive what they are, then you need to record for a very long time, because you need to look at so many of the possible input patterns to be able to deduce what's on the output. And it becomes a very complex mathematical problem. So it's mathematically complex then with a big black box. And it also requires possibly more observation than you could possibly do in an experiment or even over the lifetime of an animal, because there are aspects of the structure in there that could be innate from just the development of the brain, and aspects of its function that you never saw over the course of your recordings. So it needs to be a fairly small, limited in size, feasible black box that you're dealing with. And that's why in the end, when I talk about how to use it, it always ends up being this conversation about breaking it up into lots of little black boxes that are feasibly large. But still, yeah, tons of problems there.
So I had a question sort of related to that. So if you're trying to if you're trying to build a black box which simply behaves the way the brain does, so this is from an artificial intelligence perspective, what level of what do you think would be the level of granularity of modeling of the brain that's necessary? And more importantly, perhaps, how do you sift through and filter out parts that are just kind of evolutionary artifacts and not really could be done in a simpler way artificially, to the end of creating a black box that actually so the minimal internal complexity to produce the black box behavior?
Well, if you're asking me, and I'm guessing you are, you're asking really tough questions, and I don't really have a good answer. The granularity, I don't know. I mean, maybe we can start with something that seems to work right now, and I'll point again to some of the work that's been doing system identification at the level of action potentials. spike timing and the fact that it's been able to reproduce some useful behavior, or brain-machine interfaces where it's possible to use the spike code that comes out of the brain. But then even to figure out which parts could be reduced, could be somehow compressed to a simpler representation, what is just a result of this evolutionary patchwork that is, of course, suboptimal, I don't know. I mean, I think we have to study that way better to figure that out. It's too early.
I think perhaps one hint that might lead us in that direction is the level of units that are sort of replicated across the brain, things like cortical columns or these structures that are massively repeated in the cerebellum. They suggest to me that we might be able to get away with black boxing structures that are much larger than neurons. So we might be able to actually sort of turn these large groups of neurons into sort of a single functional unit. Now, Randal pointed out that by being a larger unit, it may have a more complex function, which is certainly valid. But it'll be interesting. I don't think we know the answer, but I have some hope that we may actually be able to get away with modeling a smaller number of these larger units. And the evidence I take from it is that the underlying sort of evolutionary path and sort of neurophysiology has itself sort of converged on this massive reuse of similar patterns, which I think may indicate that there's a lot of a redundancy at the neural and subneural level that we actually we might not need. I don't know.
I think
Following
it's
on
important.
from that.
Oh, go ahead. Go
Sorry.
ahead. No, go ahead.
Yeah, sorry. Following on from that, it seems like if we're interested, and the original question was to do with human beings, but if the most interest is in somehow virtualizing a person or something like that, are we really going to say that we need to also virtualize some peristalsis in the guts to have a successful human being? Or, I mean, I'm sure we can simplify the whole system a lot without losing what we would call still an individual person.
So I have one thought on that, actually, which is it's a really important point, that humans don't exist in a vacuum, and we evolved as a response to our environment. And we've had this notion that we could somehow extract the computational principles of the brain, and that would be sufficient to recreate even things like human experience, or consciousness, or feeling, or whatever, But increasingly, we start to look at the human, cognition and all the properties of our brain as embodied, and which means we have a body, and our body interacts with the world, and we're part of a dynamical system that extends far beyond our bodies. And this is one of the big challenges, I think, is even if we get around to mapping all the parts of the brain, we're going to need also model the context, which means the environment around that agent, to really understand, I think, what those dynamics mean. And that is a much more complicated problem. Now, it's not just one brain, it's many brains, so to speak, or at least the behavior of many parts of the system that's hard to observe. The other thing about, you mentioned a nice little point there about, do we need to model the bacteria in your gut or various other kinds of things? Well, if we look at human emotion, for instance, human emotion, we can think of like the role of, say, the insula as basically being a gut detector, picking up changes in chemical changes all throughout the body that all goes up these thalamic pathways and activates the cortex, and from that, somehow, through a layered computational system, we get this thing we call emotion. Now, if you ruled out all of those downstream inputs and you just kind of didn't track them at all, we probably would not really be able to understand what computations were being carried out in, say, the insula. So maybe we do need to actually model the whole body to fully understand what the brain's trying to do. So those are my 2 cents on it. and one thing I would say, just as a sort of systems neuroscience-y kind of person, or dynamical system person, it's very hard to partially observe a piece of a complex system that has a lot of interconnections with another part of that is not observed, and try to make inferences about the rest of the system, especially if it's a close to chaotic system or a highly nonlinear system. And so we do, because we know that the brain does receive so many inputs from the rest of the body, I think just from a purely engineering point of view, it would make sense that we need to observe a few more data points outside of just the brain itself to make sense of what's going on. Yeah, anyway.
Yeah, it makes sense that, of course, embodiment is extremely important in this context, and I think that generally we give it short shrift by not talking about it in presentations, etc., because the brain itself is such a complex issue for us to deal with. But yeah, I agree with that. At the same time, when you're thinking about, well, how do you present that whole environment? Well, one thing you can do is put your emulation in that environment, in the real environment. Don't model it. Put it in the real environment. So that's where, again, the comparison with doing neural prosthesis comes in. If you get a neural prosthesis, sorry, hard word, you have to put it in your head, right? It's in your actual head, and it's part of your body. It's part of the whole environment. So there you go. Now you can do that with a larger prosthesis, maybe all the way up to the size of the brain. Not saying that's the best way or easiest way, but that's one possibility. So yeah, I agree with that. That said, I think some of the things that we think might be particularly complicated because the body is such a huge, complex organism, when you actually look at the input-output between body and brain, I wonder if that's particularly one of those places where compression, where a reduction of the complexity is possible. I don't really know, but it's something that's interesting to look at.
I think, yeah, Tim, you raise a great point, and I think this issue of knowing the input, for example, from the body is not only an issue when you think about external inputs, but it's also an issue when you think about what's actually happening in the brain itself, and you make this point, which is great. I think we often, all of us, fall into the logical trap of thinking that when we record from a particular brain area, we see some input, we see some output, and then we try to come up with a transfer function. Whatever that transfer function is, we then reassign back to the brain area where we were recording. But of course, it's important to remember that brain area is actually communicating with hundreds of other black boxes, and those other black boxes could in fact be the site of computation. So, you know, as a field, I think it's always very important to remember as we go forward with the physiology that just because you record a signal in a particular area doesn't mean the signal is computed or created in that area. And so to really be able to separate inputs and outputs and to modularize the brain, whether it be cutting off external input or just cutting the internal systems down, I think is really going to require interventions that isolate specific components. Otherwise, it's just too complicated.
So
yeah, well, I saw Randal quoted Richard Feynman doing a little research for this, saying, you know, I cannot understand something unless I can build it. But the question is that just a necessary thing, or is that a sufficient thing? So plausibly necessary, but not sufficient. Sorry.
Yeah, it could be the other way around too. It could be that it's sufficient, but perhaps not necessary. Maybe you don't need to be able to build. Maybe building it is just the way to be sure that you know what's going on. I don't know. Could be either way.
So Randall, I have a follow-up question about what you mentioned about neuroprosthetics. As you mentioned, most of the neuroprosthetics I've seen are really invasive, like you have to implant them in someone's head. And as someone working more on fabrication and manufacturing, I was wondering what are some of the areas that we can help with in developing neuroprosthetics. So I was thinking, like, you know, there's a lot of like emotive EEG, those trade off some of the signal quality for non-invasive, but like what's the medium there? What sort of thing could we focus on for manufacturing?
I'm not sure that there is a medium. I mean, I've looked at a lot of these different tools as well, and personally, I'm not really very optimistic about things like EEG and TDCS and all of that, because really, you know, this is the problem of you've got a huge black box with billions of parameters in it, and you're trying to record just from a few data points on an EEG. When could you possibly get the resolution you need to break down your box into such a small pieces that you could actually identify what's going on in it that way? This is the reason why, you know, why we why it ends up being something invasive. And I don't know if you can get around that. So maybe the better thing to look at is how can you make these sorts of more invasive procedures safer. I mean, if you think about it, every time we take a drug, like an aspirin or something, we're also doing something invasive. We're putting something in our body. We're putting chemicals in our body. They could harm us, but we trust it because we've tested them and we've become comfortable with that. If you can build something that can get the data you need to get, and it is similarly trusted, then you're basically there. It doesn't need to be something that's external to the body, as long as it's something that a person would trust.
Okay. What I'm curious about is what's the major limitations so far with developing of like brain emulation?
I'm sorry, I'm not sure I understood. Could you repeat? I just had a
Like,
moment.
what's the major limitations in developing brain emulation?
Well, I think the main
Hello?
limitation still. Am I not audible?
Yeah,
Can you hear
I
me?
can hear you. Yep.
Okay.
We're good.
I think that the main limitation is still this combination of getting enough data, both structural and functional, and being able to do something useful with that data. I mean, even the OpenWorm project, which is just dealing with a nematode, is not finished yet, right? So this is still a really big problem. Once you can do this for a very small creature, be it a worm, be it a Drosophila, fruit fly, I think from there on, the whole process of scaling things up, that's a whole different question. That also probably has its big hurdles, but to me, they don't sound like the kind of hurdles I'm as worried about as the ones of just being able to do it on the small scale.
Okay, I missed the earlier part because my phone earplug unhooked. So you mind repeating that again for me? The major limitation?
Yeah, just in brief, the major limitation, I think, is still gathering the data from the brain and knowing what to do with that data.
Okay, is that limitation mainly due to hardware than algorithmic processes, or what was the limitations in terms of collecting the data?
Some is very basic. We just don't have a tool that you could say would just give you all of the activity data of every neuron in a brain. We just don't have a tool that does that, or
Okay,
that gives all the connections between all of the neurons in the brain. We don't have that tool either. So those tools simply aren't ready yet.
okay. The thing about it, I'm wondering if like in terms of a as a group of people interested in this area, is if we were waiting on somebody to develop the tool, or are we going to develop the tool, or necessarily, because as Kimberly somewhat pointed out, is that not necessarily that people out there are working on this particularly for us. I don't know in terms of how who's developing what, but it'll be interested to know to find out how exactly are we going to go forward in terms of collecting the data.
Yeah, and I want to augment that too. So I think that actually raises a thing, which is what is the goal of a roadmap, right? So I think Chester's question gets at, so if there are these things that are missing, if there are these gaps that exist, does the roadmap tell us exactly what the gaps are? Does the roadmap limit the space of what the gaps are? When you sit down to create the roadmap and put it out there, what is your goal, and how does a roadmap get used for the purposes of making progress?
Well, to me, the main purpose of a roadmap is to keep track of progress and to keep identifying what the gaps are and what may be the best possible solution. So I like to think in terms of what I call this objective-oriented reasoning, which means you have an objective, and that in this case is to be able to emulate a human brain. And then you need to think in terms of all of its constituent problems. And that gives you this map. And then you look at what are the possible solutions, which are the areas that people are working on, what are the possible tools or solutions they're developing, where is there something that's not being developed at all. And I would say, for example, the translation from collected data to a functional model is one of those areas where there's hardly anything being developed at all, maybe OpenWorm is kind of the exception, or those types of projects are kind of the exceptions. So the thing that I do generally is to try to figure out, if you had this problem, how would you tackle it? That's where something like system identification as a basic method comes in. Then break it up into all of its constituent problems and find out who are all the actors in this space. What are they doing? Who's interested at what level? So it ends up being this network where you have, say, core people, core researchers, some labs where the researchers are interested not just in developing their particular tool or carrying out their particular experiment, but they're actually really interested in the whole problem. They're interested in whole brain emulation ultimately. And then outside of that, you've got this shell where I'd say there are people who have similar goals, like they want to be able to do full-scale connectomics because they want to understand how the brain works, but they're not really interested in emulation or reconstructing a brain. And that's another set of people that are involved in one way or another. And then outside of that, you have further tool development or algorithm development, things that are involved in a sense that we need them, but the people there don't have any particular reason to want to be motivated to work with us. So it's partly this architecting problem of networking between various labs and people who develop problem solutions or have a problem that they need a solution for. And it's partly this matter of figuring out how could problems be solved, how can you get to the goal, and there's upkeep. You need to go over that roadmap over and over again and realize what has become the new gap given that work has happened in an area. And so I've seen this move along, for instance, from connectomics, which has gotten way better than it used to be, so that's not as big of an issue and complication anymore. Functional recording is still a bit of a huge problem, but at least there's a lot of work going on there. Platforms that you would implement on, a little bit of work so far. The algorithm issue, very big problem. And where that main gap has been changing over the years, so it's this iterative issue.
What about even going even another level out? What about the broader community and the fact that Carbon Copies is as much sort of a community organization as a sort of a roadmap organization? All the people who are interested in this but are not in the science. Do you have a clear sort of indication of what people like that can do to contribute or to promote or advance these projects?
Are you asking me, or are you asking Steve Larson? I mean, he's got the whole distributed network thing down pat, right?
Jump in.
Oh, okay. Sorry, I thought that question was mainly for Randal. So,
anyone with an answer?
Yeah. Well, so it's interesting, right? So Chester, for example, runs a AGI forum. Artificial General Intelligence Forum, and I get the impression that folks there are coming mainly from the computer science side. And I think a lot of this talk also relies on knowing, the talk that Randal presented in this discussion, also relies on knowing some of the neuroscience or enough of the neuroscience to be able to appreciate the technical details there. And of course, there are these two approaches to artificial general intelligence, one which focuses on sort of the longer path, potentially, or I don't know, I mean, I guess it's hard to say, but one that focuses on the biology and the tissue of the brain and reproducing that, and the other one, which we've seen progress on in terms of deep learning, is sort of about coming up with algorithms that process data that take inspiration from the tissue of the brain, but don't necessarily seek to reproduce it exactly. And so why do I mention that? If we're asking about this question about community and what communities can do, the first thing to understand is where your potential community members are going to be coming from. Are they going to be coming from having a knowledge on the artificial general intelligence side? They're going to be coming from a perspective of understanding the neuroscience. There are more and more people who are at the intersection between these two, but I think the set is still pretty small of folks. And so I'd say one of the missions that we think about in OpenWorm is actually education. And we really try hard not only to put out work that scientists are going to understand or neuroscientists are going to understand, but we try to put things out that really builds a bridge from essentially very basic starting knowledge all the way up to kind of where we're at. And so I would point out for anybody watching, if you clicked on the the Google Events page, that there are links to several publications that Randal has put out, and so if any of the presentation that you've seen you want to go into any more depth, I would recommend checking those out as well, carboncopies.org. But then to this audience, the question of like, how do we get a community involved? Like, it takes a lot of thought to think about, well, how could you get, Chester's AGI, forum of programmers to come in and augment the roadmap, for example? How could you, how can you reach out to neuroscientists? So from the starting perspective of somebody who's sitting there recording from the brains of mice, what would they need to know in order to be able to help contribute, to a roadmap as well? So part of it is turning it around, thinking about the people that you would potentially have in your community, and then figuring out how to educate them and how to interest them, so you have to motivate them, how to excite people to, to get into this particular, perspective, I think are all things to think about. If you're teaching people, if you're motivating people, then that's the foundation on which a lot of these communities, I think, get built.
Okay.
So, yes.
My opinion, like, there's all these separate fields. Honestly, my whole approach, at least from the way I'm approaching it, I'm trying to get grasp of all these fields. And for me, one of the limitations seems to be hardware. Like right now, in terms of what I want to do, the GPU is not really doing it. It's not really have the functionality that I want in terms of. So, and that's for gaming. So what I'm thinking of is actually going to that level of actually engineering some kind of parallel computing processor or something that because what I found is that you have all these different fields and they probably only care about what they're doing, not necessarily what you're doing, and also they have things in separate minds. It kind of limits your creativity, So for me, I'm approaching it from get all ideas in one mind, and one mind kind of sort it out, or one group of people sort it out. Try to know as much as possible from all these different fields, even though it's huge. Each field is huge. I think it's well worth the effort to like understanding engineering, understanding neuroscience, understanding computer science, understanding all these fields, so that when you look at the problem, you have these a combinatorial combination of tools to attack the problem from, not just from a computer science perspective. You can have from the neuroscience perspective, you have from the engineering perspective. So that's what I've seen pop up so far. That there's this separate fields, and they're basically not interacting with each other. And that's how I would approach it. Individuals should try to learn all these fields together.
I think that's great. I think that's a great theme, and I think folks probably have other things to say about that. But I am going to have to be mindful of time here. So thank you, Randal, and all the folks here from Carbon Copies. Thank you very much to our panelists who all got a chance to speak and weigh in on some point here. I would follow on to what Chester said about interdisciplinarity, and I would say that it's through us all creating forums like this and talking to each other and getting folks from all different parts of the question, folks that are interested in interested in doing this themselves, folks come from the computer science side, from the neuroscience side. We have an internet that beautifully allows us to connect on interest topics no matter where we are in the world, as was represented here. And I would also say, for folks that are watching, this video will be archived on YouTube, and it will be an opportunity, I think, for folks from all sorts of different disciplines to come in, see the conversation that we're having here, and be able to evolve it going forward. So I'm going to wrap this up so that Randal can go. If you were excited about this presentation, if you enjoyed this, if you made it here all the way to the end, thank you very much. Please subscribe to our YouTube channel. We will be doing another one of these in a month. We're going to be focusing back on the worm, our exciting little organism. I would also say, if you like the topic here, please visit carboncopies.org and check out additional work that's going on there. And as for the folks here joining us on Carbon Copies, I'm sure this is a great place for us to continue discussing open science, continue discussing potential ways to intersect as we go forward, and see what kind of interesting conversations and partnerships can be formed from this group going forward. So I'll wrap it here. Thank you guys so much, and let's keep the conversation going online.
Thank you.
Thanks.
All right. Bye, everybody.
Bye.
Bye.